And good afternoon, my dear students. And this is Dr. Manibalan to explain you about the structural databases for the practical session. Okay. Yesterday class we had some connectivity issue. That's why today I have making this video for you. You can go through this and you can practice it during your practical session. After practicals, we will have an G meet to clarify your doubts. Okay. In between the and if you have doubt during your practical session means you can text me. I will answer that in the group. Okay. We will see that structural databases. So, but introduction databases, as you know that databases are repositories. In that they have deposited uh, structures of biomolecules such as proteins, ligands means chemicals, RNA, DNA, everything you can get. You can get 2D and 3D structures also. Right, mostly we have used 3D structure for the analysis, and sometimes we also use 2D structures, chemical structures, which means drugs, right? And the purpose of the applications of a few applications of structural databases are we can visualize structures, molecular structures in 3D format, and you can rotate, zoom it, and you can find the amino acid positions and how they are connecting, and what is the ligand interaction between and that protein and the drug, or how what kind of bonds are made and what is the length of the bond. So everything you can get, you can zoom it, and you can just play how to see how the molecules are, whether it is alpha helix or loops or sheets or there. So as a result you can paste, you can take a snapshot of everything, you can just paste it and discuss it, right? Okay, protein. So next we can also model the structure of protein which doesn't have the validated experimental structure. Okay, so we can model a structure for a new protein. uh, for example, if you have only the sequence and the 3D structures are not yet revealed by X-ray crystallography means you can model it. For that we can have so many softwares and tools, servers are available. We will see about this modeler or modeling softwares in the separate session. Okay, the last we can understand the mechanism of molecular level by using the 3D structures of proteins and the toxins. Proteins, toxins also a protein, right? So we can easily identify the active site and what are the amino acids present in the active site, what are the volume of that, surface of that, whether it is hydrophobic or hydrophilic. We can easily analyze by using this 3D structure. By this way, we can understand the molecular mechanism of interaction between so many ligands or with so many proteins. Okay, so classification of structure databases. So we can have protein structural databases. This is core. You can get the protein X-ray crystallography structures or NMR structures also. And the second one is about protein classification databases, which means you can classify. Suppose if you want to know that in which classification your protein of interest is comes, means you can go to the scope and cat, you can find that. Previously we are using pfam, now it is updated it to interfero. So pfam also available with integrated with interfero. And this is the structured databases. And today's experiment we are going to see intensely about rcsb pdf. Shortly we can say pdb, not pdf. So pdb, protein data bank. So here PDB means Europe. Okay. So that's all about the protein structure databases and nucleic acid structures NDB and RNA central. NDB nowadays it is not working properly. I think they have some issues. Uh, as soon they can clear that. I think. Okay. You can use RNA Central to get that. After that, if you want to do analyze in the protein ligand interaction, means you can go for database binding database, Chembel, that is especially for chemicals and drugs. You can and have the appropriate target also. Likewise, drug bank also has the same like Chembel, and but here you can find the uh, properties of drugs, which means chemicals and respective targets and how it inhibits, what are the concentration, everything you can have in the drug bank. So at last, computational drug prediction databases. That like modeler, we have databases. With those databases have the predicted models. So you can submit your query, you can get the models of the proteins. Okay, for example, AlphaFold database, Modbase, and Swiss model repositories. So you can use all the three. And recently, AlphaFold database given by EMBL and Google. I think that is deep, somewhat they called it that. We will see that during the practical session. Sorry, during my explanation. Okay. Mm, and tools for analyzing structural data. In databases, you can have structures, right? Say aim, you can see that. Okay, see to retrieve 3D structure of a protein from data bank. Mostly, the PDB also has their own visualizing in that page. You can visualize 3D structure that you, but which means you can rotate and see everything in the PDB itself in the online. But if you want to extensive research, you should download any one of the viewer, right? Those viewers, my choice is Pymal, Chimera, and Discover Studio Viewer. You can have any one of them, and you can download it. You can use it, right? At present, I have Chimera. Primal also very easy. That is Python molecular viewer. There are so many tutorials, lots and lots of tutorials for all the three are available in YouTube. You can go there and you can learn things how to play with protein molecules and ligand or docking results. Okay. So at present I have camera. I will show you how it works. Then the experiment is retrieve and visualize a protein structure from PDB. So this is protein data bank. Okay, so you can have Chimera, Pymal, or Discover Studio Visualizer. Okay, protocol. You can just visit the PDB and you can search the protein in the text box, because if you are new for the PDB means you don't know the PDB ID, and you can search it from any other journal, or you can search it in Google itself. So you can get the PDB ID of your protein. Instead of that, you can directly go to PDB site. You can type your protein of interest. Don't go for this hemoglobin unless otherwise you chosen as your protein of interest, right? So you already chosen a gene, which means a protein also. So you can use that protein name to search to do all the experiment, right? So filter the search. I will show you how to filter the search in the PDB, and you can check the experimental data, validation, and structure, sequence, and ligands, and you have to paste all the results and discuss it. After that, you should download the structure of the protein, whatever you have chosen in the PDB format. After downloading, you have to visualize by using any one of the viewer, molecular viewer. Then in the during the viewing, you can um, show me how many, which means helix, what are the helix present, helix and sheets of proteins of your protein. Okay, you should say how many, what are the, you just show me that helix and sheets and the ligand if any, which means, so you can zoom it and show me if ligand present if any. in the PDB structure which you are downloaded from the PDB, and use cartoon representation for the carbon uh, entire protein. So you can use a cartoon or ball and stick model, a surface model, wire, all are available, options are available, whatever you like you can just show me. And here uh, our aim is you have to familiarize with that app, which means the tool, viewer tool. So you can color different chains. If your protein of interest have so many chains, means you can color any one of the chain with any color. You can show that and rotate and analyze the structure. In this, you can rotate it. You can see any metal ions present in that, uh, and if you zoom it and take a snapshot and produce as a result. Okay, you can analyze the amino acids numbers and the bonds between that are hydrophilic or hydrophobic amino acid or loops or whatever the structure. If you have any special in the structure means you can uh, show as a result, right? Okay, and this is about the PDB, and the next video I will show you how to classify the scope. Classify by using a COP and CATH and ProSite. Okay, ready? We will go for PDB now. So you can just PDB in the search box. You can have this RCSB PDB homepage. Just you can go here and it will show you that. Just go through this. Mostly, most of the people can use PDB for downloading the structure alone. See that there are so many uh, options are available. Sequence similarity search, structure similarity search also available, chemical similarity search available, chemical sketch tool is available. By using this tool, just click this option, you can use and you can draw your chemical, right? Ready-madely you can draw. This is Marvin sketch, I think, and they have it. So you can just go, just click it and go. You can draw the structure and you can download it in any format, chemical format. There are so many formats, small 2 format, PDB format, and smile format. You can get it, right? Okay. Then visualize. You can. They. I told you they have their own visualizer and sequence annotation viewer also, genome viewer also available here, and analyze. See that pairwise structural alignment. Yeah, this is your next experiment. I think hopefully we will see in that. So structure quality we are going to analyze in this experiment also. So download, you can download everything, sequence, ligands, files, which are all submitted by the scientist, right? So if you want to learn courses means you can have the training courses also. Okay, this is about PTB. So now we will download our PDB structure. See that what I have to do. The print procedure protocol is visit the PDB and type a protein name, right? So you can go here, protein name. Just I will give some protein name first. Hemoglobin, okay? For you? So I will just take hemoglobin. You can take your protein of interest, right? Hemoglobin, search it and see this nearly 335 pages. Each page contains 25. If you want, you can change it. How many structures will be shown like that. So, and this is the see number of structures available for hemoglobin. In this, I want to get only the experimental structures. And next one is I'm searching the structures on Homo sapiens, right? And these are the options available to filter the result. So you can use the left hand side panel to filter it. And taxonomy, so I already selected the Homo sapiens, so directly it will come to the eukaryotic, right? Okay, experimental methods, what kind of structures you need? Mostly, not mostly, you should select X-ray diffraction method, right? That is validated, that will be used. So other methods, electron microscopy, solution NMR, neutron diffraction, electron crystal. The theoretical model also is there available. They have included now, I think, theoretical models, right? So we need only the X-ray diffraction method, that is a laboratorically validated structures, so the scientists will use to further research. So you can go for resolution. So resolution means if you select 2 or less than 2 angstrom, say that angstrom is the unit of that is the distance between the eyepiece and the sample during the refinement analysis, right? Getting the structure. So you should choose less than 2 or 2 or less than 2, right? So you can go for 2. Sometimes your proteins may not contain this much resolutions, right? Don't worry about that. Whatever available you can get first, because there is no research on that particular protein means they will not mention here. So whatever, some proteins, in some cases you only have 4. What to do? For that time there is no option, so you can download that and you can use it for further research. And here we have so many options, so I have selected for 2, and more than, this is more than 2, that's okay. Less than 2, okay. So usually you should select, if available means, uh, 2 or uh, less than equal to 2 will be the best, okay. So release date, no need. If you want you can select anything. If you are searching or researching any uh, specified enzyme means, you can classify, you can check it, okay, and you can give it okay here. The same thing is available in the top of the page also. The data is loading, so see here also available, you can give your box here. This is the structure. See, now I can have only 937 structures, 967 structures. So in that I have to definitely we have the structures of less than 2 Armstrong, right? So previously you should know what is the length of your protein sequence. That will be helpful. So you can compare with this, because this will be, see, hemoglobin, if you have 967 success means how they will come. Scientists may work on mutation, scientists may work on the specific domain, the scientists will they might be work on the specific chain also. All the structures modifications will be deposited after crystallography, X-ray crystallography. So this many structures are available for you. So you can choose any one and you can get it. I'm going to choose this thing. What is that? See, crystal structure of ferrocyanide bound human hemoglobin A2 chain at 1.88 resolutions. Okay, I will check that as I will take as an example. So just you can click here to get the structure of that particular protein. See this? This is biological assembly. Here you can't rotate, right? But you can see, you should copy and paste this result, experimental data snapshot. So method is X-ray diffraction, and here you should see that R free values. R free value is here 0.195. Usually resolution divided by 10. is the good or free value, right? So 0.1 is good. So this is the value by the depositor. Okay, depositor means the scientist who have submitted the structure. So our free value is good, and here you can validate the structure. So this side, the red color side is worst, and this blue color side is best, right? Better things. So clashes, you can check that clash score, how much in an outline is. So you should know about the Ramachandran plot. I think hopefully you have already studied about Ramachandran plot, right? So there is no clashes and no outliners. So this will be a good structure. And please be careful while you are selecting the structures. And see that I told you less than 2 is better, right? Resolution at Armstrong's less than 2, because in the 2 you can get clear uh, view of that structure. That's why we chose less than 2 or less than 2 Armstrongs. So that is one of the category to select the molecule for further analysis, bioinformatic analysis and research. Okay, so you can snapshot, you can take the snapshot and produce as a result, and you can go down further, see that there will be a two chain in this structure, hemoglobin alpha chain, and this A and C's are available in this sample. Sequence length is 141. You just compare with that uniprot sequence, how many length is there of your protein. And organism, okay, mutations, there is no mutation. Sometimes you can find the mutations. Mostly working on wild type is good, unless otherwise you have specific research on mutations, right? So you can have the this also as a snapshot. You just see that reference sequences, how many available, and uniprot, everything. So you can come here and see that there will be a molecule with hemoglobin delta chain, that ID number 2, which is have B and D chains, same thing, 146 sequence length is there, right? And here ligands, there are 3 unique ligands are found, they have given with the 2D diagrams. Okay, if you want to know that interactions, you can just click it and make a snapshot, right? Focus chain, mm, GLNU with A, A, B, C, D is another chain, and this is the amino acids. Okay, and these are the ligands. There are 3 more ligands, unique ligands. That is protoporphyrin 9 containing FE, like that. And here is ferrous again ion, ferric, I think, and here is cyanide ion. Those will be in the structure. So this thing you already had in the structure itself previously, and this is the deposition detail. So in this page, you should take snapshot of unique ligands present in that, and the chains of your own protein with the sequence length, and this thing, experimental data snapshot and validation snapshot. So you have to take a snapshot as a result. Then here, come to the picture now, and you can find structure here, right? In the top, after structure summary, you can get the tab of structure. In that you can go, you can find the sequence of, and here there is a viewer. You can rotate and see how many chains are there. These dots are water molecules actually. See that? What is the polymer? So you can and go for different. This is cartoon, right? You can go for, see. So these are the ligands present in that. You can take a snapshot of that before going for PyMol or Chimera analysis. These are the ligands present in that. So water molecules and then ions. See, ions I have Okay, you can uh, you zoom by using your mouse, zoom it, you can see that where it is. So you can see the polymer, which means the structure. Again, ligands, how many ligands are there in that, and water molecules, right? So ions. So that is the only one ion, I think. So I can hide everything except I can hide everything except iron. See that these are the, I think, cyanide ion. You can get the detail of the structure. This, oh my God, what is that? Hmm. If you place here, definitely you will see that what is showing. It's not like hand, right? You just placed it. I think it was hide. Okay, and see that. Now you can see it's a hemoglobin delta B threonine and 84th amino acids. And if you go and place the cursor here, means it will say that is cyanide ion. So by this way you can take snapshot by uh, searching the polymers or ligands and water molecules. So what kind of ligands are there, you already shown, right? You already knew that. So this is Porto 4,5,9 containing ferrous is the ferric compound. So this will be shown, right? So you just zoom down and you can go and select that. See, one more molecule is there in the bottom. In the right hand side bottom, you they will display whatever the cursor is denoting, right? So this is about the structure of proteins you can get, and annotations if you want, you can everything you can have in the first page itself, I think. So no need of putting as a result these annotations that we will see this annotation in the scope. Okay, scope and cat we have in a different experiment, so no need to that experiment. Also we have seen sequence. If you want to see sequence, you can do that. Genome, so if okay, see that this is the genome, chromosomal structure and the genome. So don't worry about this. If you have, see that positions are available. Okay, so for you, as a result, you can take the results from structure summary and here experimental sequence if you want. Okay, next thing we will download the uh, files, PDB file. See that. So in the right hand side top, you can find the download files, right? And please listen carefully and see. You can have the option for FASTA sequence, PDBX, chemical format sequences, binary sequence, and PDB format also, right? And PDB is that also. And you can download anyone. So you can download PDB format. So I just click here and I can have that. See PDB. If we have viewer already, means it will display like that, right? You can find that .pdb. The .pdb files will be opened by using text editor. Okay, and see that. I will show you downloads. Here today is there, right? That is .pdb documents. So I can just open it. It will ask how to open. It cannot read because I have selected the wrong app defaultly. Okay, how to close this? So I will right click and open. Probably you don't have this problem because I have already assigned this tool to open that, because this is a book opening tool like Adobe Reader, right? It's defaultly it opens. So you can go there and open with Notepad. And this is the app, the Calibri ebook viewer. It's defaultly it open, but it can't open that. It has set it like that. Okay, so the Notepad, you can choose Notepad or text editor. I am using Notepad here to open that. See, this is the PDB file of that protein. So these are all informations about the chains and experimental data. See, you just come to this part. Okay, this is the amino acids and the heme protein and containing ferrous are there. And everything is here. And these are the say that atom. This is what exactly we need to study, right? These are the coordination and the x, y, z coordination of each and every atoms of the protein. Okay, this is the atom number they have given, and they have given the chemical type and the amino acids and the atoms and what are the position of that each and every atom in that. Okay, so by this way they have given as a digital one. This file will give a 3D molecule, 3D view in the viewer, right? So just we will see that how it will get opened. See, so we will close it and I will open that. That is called as 1SHR, right? So in my system I have Chimera 1.9. You also you can download and view it. See, and this is how it will open. You can go file, open, and better you can download the structure in the folder which you have already created in your name, right? Which has sequences already you have put, I think. So you can go that, you can go for download and I will open it. See, this is how Chimera will open. You can zoom it. You can see that how it is. Okay, you just take a snapshot by using this. So um, I will create another video how to use this Chimera for your own protein or otherwise this protein. Okay, so you can take a snapshot here itself. That's why okay, we'll make a separate video. I will send you.